Alright, the final lesson that we're going to talk about and the final topic of Unit 3, Area Study 1 Bio, we're going to finish off cellular respiration. We want to really talk about the whole biochemical process of aerobic cellular respiration this time, and that involves talking about cycles and chains, but not cycles that are going to be attached to a railing like this, even though we are going to talk about this guy, the Krebs cycle. So really quickly pause the video here and we'll have a look at the 3 quick questions. So the reaction ADP plus PI, which is a phosphate ion, becomes ATP, right? Is it irreversible? Does it occur without the presence of enzymes? Occurs in yeast cells during fermentation? Only occurs in cells containing mitochondria? Hopefully you saw that the answer is C, okay? Because that's one of the things that happens in fermentation. We get the production of ATP. Below we have a diagram over here that shows the structure of a particular polypeptide sequence. The protein actually contains 2 distinctive types of polypeptide chains, highlighted in red and yellow. What is the name of these 2 types? And hopefully you can see that here we have like a helical type structure, so that is a alpha. And we can sometimes write alpha like this, helix. And then the yellow one is a beta, and we can write beta like this, pleated sheet, right? Because it looks like a sheet. The polypeptide sequence can be accurately labeled as a protein molecule and is ready to take on its role within the body. In terms of the molecular nature of the protein, what level of structure must it have? So if we've labeled it a protein, it must either be 3rd or 4th, all right? But there's no indication that there's more than one polypeptide chain here. Actually, we can see that no, we can't, but it's just the one polypeptide chain, so that makes it tertiary. Okay, if it has a function, it has to be at least tertiary. Okay, in terms of the molecular nature of this protein, it has a 3D shape. Okay. Alright, so in your study designs today, we're going to finish off cellular respiration. We're going to look at the main inputs and outputs of what we call the Krebs or citric acid cycle, okay, and the inputs and outputs of the electron transport chain. And we're going to have a quick chat about ATP yield. That's a bit of a contentious topic though. Remember, details of the actual biochemical pathways aren't required. Alright, and then lastly, we're going to touch on the factors that affect the rate of cellular respiration, including temperature, glucose availability, and oxygen concentration. We're not going to go into these 2 in depth though, because we've had a good idea about them. So what's the equation for aerobic respiration? Alright, so aerobic cellular respiration can be summarized by, okay, hopefully you know that the word equation is glucose. If we're aerobic, we're going to have oxygen, right? And that glucose and oxygen in the mitochondria will become carbon dioxide, and water, and energy. Okay. If we were to summarize this in a chemical equation, okay, we'd go glucose C6H12O6 plus oxygen is O2 becomes carbon dioxide CO2 plus H2O plus, we'll say anywhere between 30 to 38 ATP, and we'll talk about that later. We need some numbers in front of these, alright? So if we have 6 carbons on this side and just 1 carbon on this side, that means we need 6 carbon dioxides, okay? We've got 12 hydrogens here, but only 2 hydrogens over here. That means we need 6 of these, right? But now we have 12 oxygens over here plus another 6 oxygens makes 18 oxygens. And we already have 6 oxygens on this side, so we need another 12, so we're going to need another 6 oxygens over here. Alright, good idea to know how to balance that equation, because for an example there, I didn't know off the top of my head, I forgot it, but by balancing it, I was able to actually work it out. So aerobic cellular respiration can be broken up into 3 separate steps, and each is actually dependent on the products of the previous steps to actually undergo its own reactions. We've talked about glycolysis. We've had glucose turn into pyruvic acid or pyruvate. That pyruvate goes into the Krebs cycle. Right, the results of the Krebs cycle are going to then make sure electron transport chain actually undergoes and produces a heap load of ATP. Okay. So let's revise glycolysis really quick. It occurs in the cytoplasm of cells and yields 2 ATP and 2 NADH molecules. Okay, so we break down glucose into pyruvic acid, forming those 2 ATPs and 2 NADH molecules. The 2 pyruvate produced are going to further be broken down, so these are going to go into the next stage, the Krebs cycle. All right. If in a eukaryotic cell all of these products, if we were in a eukaryotic cell, all of these products are going to be transported into the mitochondria through active transport, and that actually requires 2 ATP molecules. So we can sort of say see you later to these 2 ATP molecules we originally produced, and that's why we're starting to get a bit of alteration to our ATP. The next step is the Krebs citric acid cycle. Doesn't matter which one you call it. It actually occurs in the matrix, so that's the cytoplasm of the mitochondria, and we're just simply further breaking down pyruvate, and we're going to form 3 carbon dioxide molecules for each of those pyruvates formed. Right, so pretty much if we have this C3, means that's the pyruvate. Right, the C3 is going to be first broken down into C2, and here we have 1 carbon dioxide, and then the pyruvate is gonna that carbon molecule is going to be further altered and broken down, and here we have another one, so that's the second carbon dioxide. We're going to get a little bit of ATP coming out. We're going to get some coenzymes, NADH, some other coenzymes such as FADH2, but lastly we're going to get this third carbon dioxide, because for every glucose molecules we actually get 2 pyruvates. There is where your 6 carbon dioxides are coming from. Okay. These coenzymes, NADH, FADH2, they carry energy in the form of electrons to the electron transport chain. And this is really important because the electron transport chain occurs in the cristae of the mitochondria. Okay, so this is the cristae here. Okay, and the cristae, what happens is these loaded coenzymes actually provide energy in the form of electrons to membrane enzymes, so membrane-bound proteins, which will pump protons into the inner membrane. So stripped from these NADH and FADH2 molecules, these electrons are going to provide energy to the proteins in the membrane, and hydrogen protons are going to run through these membranes into the intermembrane space here. Okay. These electrons actually run down these enzymes, as you can see here, and then finally at the end they're actually accepted by that oxygen. All right, this is where the oxygen comes in to cellular respiration, and by being accepted by the oxygen, we're getting the formation of the water. Okay, all of the protons that are in that inner membrane, in up here, all right, now are really highly concentrated up here, so they're going to come rushing back through another membrane-bound enzyme called ATP synthase, right? And then when they actually rush back into through the cristae membrane, this is actually providing the energy needed to turn ADP into the ATP we want, right? Here's where we get that bulk amount of ATP from cellular respiration, okay? Like I said, this process is very complicated, but this is the biochemical process. You don't have to know all of this, but if you have an understanding of it, you might find it easier to understand where we're getting this ATP from, where we're getting oxygen, and where is oxygen, where we're getting water, and where is oxygen being used up, right? What is the purpose of this NADH and FADH2, okay? So you don't have to know the specific details I'm explaining to you, but it does help to understand these other things. Overall, the inputs and outputs can be visualized here. So the first input of cellular respiration is glucose. So inside of the cytoplasm, we have glycolysis happen, so the breakdown of glucose. We get 2 ATP out of this. We get some loaded NADH coenzymes, okay, and we also get 2 molecules of pyruvate, which actually get used up in the Krebs cycle, which happens in the mitochondrial matrix, and we get 6 carbon dioxide coming out of this. We also get a tiny little bit of ATP, but that actually gets used up by the cell. And we get further NADH. Okay, that NADH is going to provide the energy for the electron transport chain, all right, and we also have an input of oxygen, which gives us an output of water. That's where those protons go. And finally, here we get a bulk ATP yield. Okay, so in terms of ATP yield, there's a bit of an issue with this because glycolysis always gives us 2, Krebs or citric acid cycle always gives us 2, but the electron transport chain, we don't always get 32 out of this, okay? We don't have, this is in absolutely perfect lab conditions. In real life, we're not going to get that. We might get 28, we might even get 26, okay? So depending on the situation, we're not going to have the full 38 molecules of ATP being produced. We might only have 30 molecules, for example. So in terms of factors that actually affect this process, there's different factors which can limit the process and increase or decrease the rate of cellular respiration. One of these is temperature, glucose concentration also, and oxygen concentration. I'm not going to discuss with you guys these. I want you to actually propose a hypothesis for each one of these and have a chat to each other about how each of these things might affect the rate of cellular respiration.